We'll now start to take a more detailed look at some of the more common markers that are used in molecular ecology studies, and we will start in this video with microsatellites. And just as a reminder before we get into this whole field of molecular ecology relies on really two kinds of data sets, and that's what I really like about this field and think is so interesting, is that it's this merging of two different things. But we need both molecular data sets and ecological data sets. So ecological data sets are things like looking at growth, so how this particular fungus, I think that's a fungus, I'm not sure, how it grows differently in different environments. That would be an ecological data set, or what the temperatures are in different environments. And then really what molecular ecology does is combine that with molecular data sets, so with the DNA or RNA. And so in this module, we're focusing on molecular markers that can be used to contribute to this molecular data set aspect. But remember that really for molecular ecology studies, we really need both of these types of data sets. And just to clarify any potential confusion right off the bat, microsatellites, which are often abbreviated microsat, or you might even see it like this because that's the chemistry notation for micro. So microsat, SSR, STR, those are all different names for the same thing. So microsatellites, SSR, short tandem repeats, that's what STR stands for. Those are all the same thing. So anytime you see any one of those terms, this is what they're referring to. And what these microsatellites are, they're these tandem repeats of 1 to 6 base pairs. So in this image here, each of these loci is a different microsatellite. So here we're looking at examples of 3 different microsatellites. And like we said, they're these tandem repeats of base pairs. So at locus 1 here, this is a TA repeat. And if we look, it's repeated 1, 2, 3, 4 times. So the way that we would write that is we have that TA sequence, and that's repeated 4 times. And for this locus, so we have 2 different strands of DNA that are shown here. That's to represent this biparental inheritance. So these particular microsatellites would be found in nuclear markers, because remember, nuclear is biparentally inherited, where you get 1 allele from each parent. And so one of the benefits of microsatellites compared to some of the older markers that we skipped, but they're mentioned in your textbook, is that you can differentiate heterozygotes and homozygotes because you're able to look at both alleles. So in this case, for locus 1, this individual is a homozygote because they're the same number of repeats on both chromosomes. And that's really the main thing that we're looking at here in microsatellites, is how many repeats there are. So that's what we're concerned with, is essentially what is the length of this particular microsatellite and how is it different either within an organism on these different chromosomes or between different individuals. So in this example here for locus 2, that microsatellite is a repeat of the TAA nucleotides, and for this first allele shown here, we have 1, 2, 3, 4, 5, 6, 7, 8 repeats, whereas here on the bottom, that's only 7 repeats. So this would be considered a heterozygote because it's different on the 2 different alleles. But overall, what we're looking at here are these repeats of base pairs, and that's what a microsatellite is. Just like with PCR we talked about before, primers are also used to study microsatellites. These black bars that are shown here in between those microsatellites, that is where you would have a primer binding to amplify this particular region we're interested in. Microsatellites can be used to calculate allele frequencies. So like we just showed here on the last slide, we see both of these different alleles. You can get information. And so unlike some of the older markers that again we skipped, microsatellites can be used to calculate allele frequencies, which is going to be really useful when we talk about some applications later on. Microsatellites also mutate really rapidly. And so what that means is if you are interested in investigating differences between organisms that are really distantly related, like this Isle Royale wolf I talked about before, and my dog Enzo, so wolves and dogs are pretty distantly related. Microsatellites are not good for studying those distant relationships because they mutate so rapidly. However, they are good for, you can maybe hear my dog Enzo in the background. Microsatellites are good for studying individuals of the same species. So I need to check my notes because I've never heard of this before, but this is a Pyrenean desman, a quote, small semi-aquatic mammal endemic to the Pyrenean Mountains and the northern Iberian Peninsula. So the overall point of this slide here is that microsatellites are not good for these distant relationships, but they are good when you're looking at individuals of the same species. And to use those Pyrenean desmans, again for an example, highly variable microsatellites can really provide some new insights for us. So with this particular species, they've done different types of studies. They have, in some cases, used mitochondrial DNA, which is, in relative terms, considered a low variability marker. And what they found when they used mitochondrial DNA was that all of these individuals are the same. They've also, though, done some studies with these little semi-aquatic rodents, mammals, using microsatellites, which, as we said, are considered highly variable. And so that high variability is good because it allows us to differentiate better between individuals. And so when they studied these same individuals, same populations using microsatellites instead of mitochondrial DNA, they actually found that there were 3 distinct populations of these guys in different geographic regions. And they were only able to identify that because of the really high variability of these microsatellite markers. So that's one, like I said, microsatellites can potentially provide new insights if you're looking at individuals of the same population and you're using a microsatellite that's more highly variable compared to something like mitochondrial DNA, which has relatively low variability.